Yeah, I mean, antibody boron conjugates, we believe, are the future of targeted drugs for boron neutron capture therapy. Simply because, as I mentioned, the drugs that are available at the moment, and BPA is approved in Japan for treating head and neck cancer. So there's quite a bit of success with BPA, but it's passively targeted. It relies on that concentration gradient to get it into cells. Antibodies, in contrast, once they find its target, they stick to the target. They're often internalized with the target and recycled. So if you can take an antibody, which is quite a large molecule, it's 150,000 Daltons, and decorate it with boron, you have the potential to use the antibody to deliver large amounts of boron to the tumor cell, in contrast to these small molecules, which boronophenylalanine, for instance, only has one boron atom per molecule. So the antibodies, we think, are a game changer both in targeting and boron delivery potential. And the reason that we're focused on antibody boron conjugates, we call them ABCs, is because most of my team here, we have 14 scientists working in the drug development part of TAE Life Sciences. Most of them come from an antibody drug conjugate background. And you know, if you've been following the antibody field, that antibody drug conjugates are a class of antibody-based drugs which have a high degree of success in the clinic at the moment, with almost 10 antibody drug conjugates being approved for treatment. So there's a high degree of success. We know quite a lot about how to produce and make antibody drug conjugates. And so we're trying to apply that knowledge to the antibody boron conjugate field. It's not a slam dunk in that the chemistry for boron is very different from the chemistry used for making antibody drug conjugates. But once we develop what we call a platform technology, which is a way of attaching boron to these antibodies, we think we'll be able to use these antibodies to deliver large amounts of boron to tumors. And one particular target that we are interested in, as are other groups, is EGF receptor, epidermal growth factor receptor. And in terms of brain cancers, if you compare expression of epidermal growth factor receptor to LAT1, for instance, it's an order of magnitude higher in terms of its expression level. So you can imagine that if you can deliver an antibody boron conjugate to a glioblastoma tumor expressing EGF receptor, then you're going to be able to deliver high amounts of boron to that tumor. The challenge, of course, in using antibodies as a delivery vehicle for brain tumors is that they have to cross the blood-brain barrier. Blood-brain barrier stops most molecules from actually crossing into the brain. It protects the brain from substances which could otherwise be damaging to it, either chemical substances or immune-related substances. And so, in addition to developing antibody boron conjugates for glioblastoma, we have to marry that to some sort of technology where we enable that antibody to cross efficiently across the blood-brain barrier. In glioblastoma, all glioblastoma tumors have their own blood supply. It's often not that well formed, and the vascular epithelium for glioblastoma might not be as intact as in normal brains, so the blood-brain barrier may be compromised, but it's not predictive enough to ensure that if you were to treat with an antibody boron conjugate, you would get enough antibody to the brain. And so having a technology that actually facilitates transcytosis across the blood-brain barrier to enable antibody boron conjugate delivery would be an advantage rather than relying on a leaky blood-brain barrier in glioblastoma.